around the vessel. This is leakage of albumin in the cortex, indicating that there is disruption of blood-brain barrier breakdown. Albumin means you have no extra tracer. In blood you have lots of albumin, and that is leaking by itself. And here is the neuronal damages. When we have given this TiO2 level cerebralizing, you can see albumin, it almost disappeared. Astrocytic activation is non-existent, and the vessel is probably the same, but it is difficult to localize. But the neurons were quite healthy, some of them. Then to prove our hypothesis, we also went at the electron microscopic level. And here you can see we have studied myelin vesiculation. ABP infusion resulted in myelin damage. These are myelin, and you know that this is white spaces means edema formation. Cerebralizing here is given nanowired, and here is normal cerebralizing. This picture is definitely better than this one. We have still some myelin preservation here, but in this case we have large number of myelinated reactions preserved. Of course, there are damages too. Then we are also discussing some of our projects with another Nobel laureate in chemistry, Sir Proto, and we had some presentation in one of the Nano Medicine Congress in San Francisco, and he was there. He really appreciated our work, so we are very much encouraged. And then the idea is, I told you, different modes of nano delivery. So the question is that we have delivered this drug using titanium nanowires. The common method is PLGA level delivery of drugs. This is done in my friend's laboratory, Giovanni Tosi, in University of Modena. We have published this paper here. So we compare PLGA level cerebralizing and TiO2 nanowire delivered cerebralizing in one example of animals. This shows that first that you see its stability, and you can see that the drug is quite stable with PLGA level even for a long time. And here you can see this Alzheimer's disease. This is leakage of radioiodine in different areas of the brain. This is in control Alzheimer cases, various areas showing high level of increased extravasation of radioiodine. Then we have this TiO2 cerebralizing here. This leakage of blood-brain barrier is significantly reduced. They are all on the same scale. PLG level cerebralizing was also quite effective, but if we compare, we can see that TiO2 level cerebralizing appears to be more effective than PLGA if we compare. But compared to the control, both deliveries were quite good. Then I show you some of the examples in spinal injury, because this is also very common in soldiers and military. And of course, we cannot mimic the human cases of spinal cord injury due to simple observations. Whenever spinal cord injury occurs in humans by a motor vehicle accident, the damage is towards the ventral side, and damage occurs when we are conscious. But what we do in animals, we anesthetize them first, and we make a spinal cord injury on the dorsal side. So point is that we cannot compare, but still we can have some data on the effects. This is also true with head injury. So we have another model of head injury. Here is the clinical situation. It's a blunt head injury, like the butt of the rifle or any kind of thing when the skull is not damaged. You have the shaky brain inside, and that is called counter coup, and the brain is severely damaged here. We have a model in animals, in rats, when this weight is dropped over this skull is not fractured, and it can produce severe injury. The proof that this model is working and counter coup, when we have weight dropped on the right side, the left hemisphere is swollen much more because the brain is moving towards that, and then we have much more swelling on the left side. Here is the example here that this is uninjured side showing much more damage than the right side. At the electron microscopy level also, the left side is showing much more damage than the right side. I can tell you here that very small changes in the environment can influence greater degree of brain injury. This is an example taken from the temperature. If the injury is occurring at cold or hot environment, you can see here at 5 degrees Celsius injury was done, and 34 degrees Celsius, 23 is normal. You can see in both cases the injury was much more severe. So the small changes in temperature can affect. To further consolidate or understand our experimental model or its efficacy, Chinese government approached us and they wanted to study this titanium nanowire delivery of their compound, herbal compound, that is well known for the treatment in stroke. So this project we have taken and presented in Society for Neuroscience in 2014. It's also a collaboration with various organizations. This is my friend Dr. Fong at the Bethune International Peace Army Hospital in Shijiazhuang. We are collaborating with that. So the concussive head injury they have chosen, and this is the 24-hour left side, right side. Left side is much more damaging. They think that this compound 60 milligram is quite good. We have given at this time. When we have given nanowire delivery of even 40 milligram of this compound, we could achieve better neuroprotection than the 60 milligram of the unlabeled compound given in identical situations. These are some examples showing blood pressure and edema given by this nanowire level. And also here showing this nano level therapy. So we can say that nanowire delivery of NDP or whatever the compound, if it is effective, its effects are much more potentiated. And here is the case of brain pathology again, and you can see that when we have treated at different time level, there is different cases of damage. And I can show you the same effect occurring to cerebrolizing. Cerebrolizing was given 4 to 12 hours after closed head injury, and in this case 30 minutes to 4 hours we can have better neuroprotection. Of course, I would like to mention that these were given as nanowire delivery. So we can say that external temperatures, cold or hot, environment pollutants, and nanoparticles can affect all these things. I'm not going to show another thing, hypertension or even drugs of abuse, because these are new trends in NIDA, that if people are taking different kinds of psychostimulants like methamphetamine, cocaine, and brain injury, what is happening. These, I have no time to tell you. And I can brief you briefly here that why we failed in clinical cases, because there are no two similar clinical cases. People are coming with different type of different disease, having taken different drugs their lives, and so it is very difficult to find two similar cases in clinical situation. That is one of the important factors. And this is Dr. Shelley. We are working with him, got Physiology and Medicine Nobel Prize in 1977, and he lives in Florida, and he is given Nobel Prize for discovery of corticotrophin releasing factors, a small peptide. And he thinks that if we use nanowired CRF, maybe we could have better effect in even Alzheimer's disease. But these results are still confidential. So he's working on this, and we are using in some of the aspects. We have produced a volume together with my wife Arna here, Recent Perspective of Neuroprotection and Nanoneurotoxicity in CNS and Neurological Disorders Drug Target in 2012. And these great neuroscientists have written comments on there. So we feel that we are doing really, in the right direction our results are going. And finally, I will conclude some of the examples in spinal cord injury. And obviously, this is a great project, it costs lots of money, so we have support from US government and even forward. democratic governance. The war injury and spinal cord is quite common, and the problem is that when a soldier gets a spinal cord injury in the battlefield, it's very less time to transport outside because we're already going war, so we have to provide immediately some relief till they are transported to specialized hospitals. So one of the aims of our project is to apply something topically over the spinal cord, so that when they are transported by helicopter or something like that for 3 hours, we try to stop the pathological process, not stop but slow down the pathological process, so that specialized treatments can be given later. So we have a model. We try to understand the deletion of one spinal cord, dorsal horn, because we wanted to see the drug effect, what is happening on the contralateral side of the spinal cord. Because any drug can be better on the non-injured material than the injured. Many cases, if you have injured the whole spinal cord, no drug is working. So we have this spinal cord, and when we have already treated then with silica, you can see that the volume expansion is much more higher than the normal healthy animals given the spinal cord injury. In this situation, this is the silica-treated spinal cord, and this ventral horn, almost complete tissue loss you can see here, as compared to this normal saline-treated spinal cord after identical injury. Then I told you of different drugs. We have given, these are from Acure company in Uppsala. They have developed potential of drugs. I am not allowed to tell the names, but you can see that they have given fixed name, SiO2, SiO1 or something like that. So we use 3 different kinds of drugs from Acure company, and we have nanowired them. The idea is that to apply to see that all drugs after nanowiring, they are quite effective, or there is a difference as well. And this is the analysis provided by the company, and you can see that the normal and laminar rats were at this compartment. Untreated were in this compartment. What you can see that this SCIO2 plus nanoparticle given 5 minutes after spinal cord injury over the spinal cord, or even repeated spinal cord here, they are belonging to this section. So they are very effective in reducing in this towards this compartment. But on the other hand, this SiO2-3 or other drugs given, they have the trend to go into this compartment, means very high injury. So I can show you with this demonstration, not all drugs can be made effective just by nano-leveling. We have to find the good drugs to nano-level to enhance their capacity. This is the morphological examinations, and you can see that this is the drug given. It is showing much more beneficial effect at the light and electron microscopy after spinal cord injury than the untreated group. So we can say that we have potential therapeutic advances using nano delivery of the compounds, but we must understand its role in inducing neurotoxicity as well. There should be a balance between neuroprotection and neurotoxicity. Either we are using nanoparticle, delivered drug, or any drug in this way. And that is the way we are at the moment working. And obviously, Swedish government is also supporting our work because these are very highly expensive materials. This is our former Prime Minister now. And I think I stop.
Yeah, thank you. Thanks for this huge presentation, because it covers a lot of issues. And I see this is also difficult to summarize 30 years in a short presentation, but what you are addressed today is the fact that nanotechnology can help or also to be as an addition to the nano delivery, because you have underline that, stress that there is not a miracle in the nano delivery, but it could be the right choice with the right drug. Okay?
Sure.
So now is the presentation is open to questions. If you have something to ask to Professor.
Yes. Thank you for the comprehensive overview. I was just wondering, you mentioned that embryonic stem cells.
Speak up, otherwise the people will
Okay. So you mentioned that embryonic stem cells adhere more efficiently to nanowires.
Yes.
So have you tried endothelial cells, as indeed, as you've mentioned also that the role is underestimated? So do nanowires help penetration of the blood-brain barrier if you use endothelial cells?
If we use nanowire delivery of the drugs, it can have better penetration into the central nervous system. And that can be found by the analysis of the same drug concentration in different areas of the brain or spinal cord after delivery of certain hours. And we have an unpublished observations. On that basis, I can say that yes, when we use this nanowire delivery or even PLGA label delivery, we can find higher concentration of the drug in the brain after the treatment as compared to the normal drug alone.
But you haven't tried cells?
We have tried mesenchymal stem cells given nano delivery. This paper is published in 2015 in CNS Neurology Drug Targets. And what we can see that we're trying to modify the diabetic-induced silicosis, And we have seen that when we have given a mixture of nanowire delivery of stem cells, it has getting good results. And recently in this year, we have shown in Society for Neuroscience that when we combine nanowire delivery of cerebrolysine and stem cells, mesenchymal stem cells, also nanowire, we are getting better results in some of the models of Alzheimer's disease, particularly on the modulation of neprilysin, a protein in Alzheimer's disease. And I have pleasure to tell you that this innovation has been just accepted for 2016 National Innovation Summit in Washington, D.C. So we are very happy and so forth.
And are you able to track them inside the CNS?
What? The cells?
No, the nanowires. The stem cells you can't track.
Yeah.
But I mean, if
There are techniques to track these nanowires using ionic microscopy, but we have not done yet. Yes.
Fate una domanda ad voce di sentore. La domanda ad alta voce di sentore.
So can you compare nanowires to carbon nanotubes which have been used in a similar way for generation, and especially the advantages and disadvantages of subministration?
This is a very good question, and I am just coming to this. We have tried single-wall carbon nanotubes and multiple-wall carbon nanotubes for administration of drugs, just as our baseline. But what we found in the initial, together with our colleague from US FDA, that carbon nanotube itself produces pathology in the brain. And we have a short communication accepted in Society for Neuroscience in 2014. And now we are working that what is the concentration, and it's well known that the single wall carbon nanotubes can damage lung, but we have seen neurotoxicity. So we are trying to study more that what is the level that can carbon nanotube, which drug can be administered safely. But we are using titanium oxide nanowire. We have even used in our laboratory by ourselves, and people in Germany showed independently that in mice even very high dose of these titanium nanowires were injected, or that titanium itself injected. They do not produce brain pathology. So that's why at the moment we are using. But even gold nanowires or gold nanoparticles, these experiments are under investigation. Since we have already submitted our abstract to some of the organizations, I am happy to tell you that even gold nanoparticles in small amounts produces minor damages in the brain. So we have to compare the different models, and carbon nanotubes are producing rather high degree of damage in small doses. That is our observation. Yes.
Thank you.
Prima c'è lui, vai.
Thanks for the presentation. Do you have an idea on the mechanism behind the improved effects of using the delivery of nanowire functionalized materials? Meaning that nanowire titanium, how does it work on the cells of the brain blood barrier? Meaning that which is the mechanism behind the improved delivery of the drug? It is just because it works as a physical carrier, and so you have more bioavailability of the molecule inside, or maybe it can work also as a physical stuff inside the central nervous system to improve as a scaffold for regeneration of the neurites and the dendrites and so on.
Very good question. I can tell you in general all nano delivery, including this nanowire. If nanowire titanium is given alone without any drug, not very different from the injured, and so we can say that this nanowire itself has very minimal effect. They do not produce neurotoxicity also. That's one thing. Second thing,
without involvement of specific receptors on
No.
the brain barrier.
if we just give titanium nanowire, the base material on which the drug was developed, without drug, it does not, neither neurotoxicity nor neuroprotection. So itself, it is not doing anything. But in general, this question is valid for all nano deliveries. What we believe that a drug is given with nano delivery, either titanium nanowires or PLG, whatever. First, that the binding of the drug is very important, and maybe it is working as slow released compound, because when it is entering, either be applied also orally topically, even given intravenously, it can reach and may release slowly these drugs like a micro infusion pump inside the brain. The second thing, its degradation is also stopped. So we can say that the drug has long time to effect and continuous release with less degradation. That could be one possible explanation. The second possible explanation is that it has widespread penetration than the normal drug because of the nano delivery, because we have seen the concentration measured in different areas of the brain. But apart from that, it is still speculative for all kinds of nano delivery. These are the two possible aspects on which we are working at the moment.
Okay. In terms of nanoprotection and neuroprotection of nanowire system, do you have any evidence of modulation of the synaptic activity of the intrinsic properties of neuron in terms of firing frequency and some modification of the activity of the nervous circuitry?
We have not used any immunocytochemical studies of synaptic function, but we have examined at the electron microscopy on those areas where we get maximum neuroprotection. What we can see that these synaptic functions anatomically is well preserved, and also the neuron glial interaction and neuron endothelial cell interaction is quite preserved. But of course, we have not used any biochemical method or immunohistochemical methods to identify.
They need a
Sure.
Yeah, sure.
Sure.
Electrophysiologist. Yeah.
The question was quite
C'è qualche altra domanda?
Yes.
You mentioned in the experiment with the study of the three drugs, the clinical drugs, that the nano leveling of any type of drug on a nanoparticle does not necessarily work. Have you made any observations on maybe some properties or size of drugs or anything that maybe would make one drug a better potential than another?
This depends on the pharmacological industry, but what I can tell you that the drug that is best acting from the Swedish company Ecure Pharma, the CIO23, it has an affinity of melanocortin 4 receptor, and maybe it can also have an inverse agonist of melanocortin 3. And these properties may not be shared 100% by the other drugs. So you can see that even a same drug, if it is working on the same kind of receptors, might be acting in a different way. Because this is a pharmacological example, serotonin 2 receptor, ritonserine, ketanserine. They are working on the same receptor. But there are lots of papers, pharmacological papers, that ritonserine is more powerful than ketanserine. The same thing happened with cimetidine and ranitidine. Both are histamine H2 receptor antagonists, but ranitidine has superior effects than cimetidine in many observations. So these are the molecular configuration. So I believe that also if we nano level ranitidine or ketanserine, they may have better effect than ketanserine or cimetidine. But this is the pharmacochemical properties on which I do not have any comment.
And in terms of elimination of the drugs and half-life, have you done any studies that show for elimination of nanoparticles in the system?
Nanoparticles can stuck into the brain or the tissues, and they have long time to stay. That's the observations now so far. I have not done that myself. I know that in Arkansas they are using this nanowire level drugs, putting in the knee surgery and close the wound, and they have faster recovery of the joint or something like that. And there is no need of these titanium nanowire to take it out. So, but I believe that they are living for a long time, and this is innocuous. That's the only I can say.
So you can predict some type of accumulation of nanoparticles if it's a long-term.
I believe that the drugs are bound to these nanowires. They can stay longer, and maybe they can resist this enzymatic degradation. So that could be staying there and having a slow release of the compound. That could be one of the ideas, but so far we cannot establish anything scientific.
Other one, I have a question.
Yes, please.
Is that because you started your speech by underlying the importance of toxicity, and also the fact that some of these compounds, not only like asbestos, have a long-term toxicity, which is very difficult to investigate in animal models or cellular models. But then, when you spoke about nanowires, titanium, maybe that you didn't mention the fact that these are biodegradable, which is their fate in the body. Because of course, I agree with you that important is the delivery of the drug, but what about the vector?
Yeah, so far my knowledge is concerned, titanium oxide nanowires are not biodegradable. But it is used in clinical conditions in Arkansas and using in human cases in knee operation and other cases. So I don't believe or I don't have any information so far. My friend from Arkansas Antian could comment on this, but this is not biodegradable. But I can only say that these are innocuous. Of course, everything toxicity depends on the time and dose. So these experiments we have not done. But USFDA allowed to have using knee transplantation in other situations. So I believe that its efficacy for a certain time must have been evaluated. I can
Other questions? Otherwise, thank you again.
It's a pleasure.
Thank you. तुम मेरा भी तो बना लो जरा सा।
हमारा बनाइए।